Welcome, biology students. This is Miss Tesselin. This is part 2 of the Neuron Online Lab. This is where we get to activate our brain cells. So before we were looking at our brain cells at rest, this time we're going to activate them. And so in particular, we're going to get a close-up of this one brain cell and see what happens when it's activated. We're going to go back to our checklist and make sure we do all the steps here. You're going to reset. You're going to make sure it's at normal. You're going to unclick all ions. Then you're going to zoom in, and then you're going to stimulate the neuron. As you stimulate the neuron, you're going to be activating the action potential phase, and you're going to watch what happens with the potassium and sodium ions. And you're going to press this a few times to see what happens. Then we're going to answer questions 1 and 2. So let's do that. So we hit reset, we unclick all ions, it's at normal. We're going to zoom in just so we can have a look, and we're going to stimulate the neuron. There we go. All right, look at that. Look at what's happening. All those ions moving in and out. Now, it says to press this a few times, but as you can see, this button I can't press it right now. So we kind of have to wait. Oh, there we go. And I can press it again. And then the same thing. So that's when we activate our brain cells. That's kind of mimicking what happens. Let's go to our worksheet, and we're going to answer those first 2 questions. How do the movements of sodium and potassium change when we activate the brain cell versus the resting? And then after you stimulate the neuron, the brain cell, why do you have to wait for it to be stimulated again? Okay. Then we're going to go to the next part. Moving on. This time we're going to be looking at some graphs. We're going to hit clear. We're going to click charges concentration. We're going to stimulate the neuron again. We're going to look at it at its peak. You can pause it there if you want. You're going to play it again. And then you're going to wait till it goes all the way to the end, where it's at the end of the action potential. And then we're going to answer questions 3 and 4. So let's do that. All right. We're going to hit clear. We're going to stimulate our neuron. Okay, and then we're going to wait till it goes all the way down, and we're going to see what happens. And eventually, actually, it starts to come back up again, and the whole thing can be repeated again once it gets reset, and then this button turns orange again. Okay, so moving back to where we said, at the low end, at the very end of the action potential, these are the numbers that you're going to record for question 3. And then, so record them there, and then you're going to answer as you compare the resting numbers with the action at the end of the action numbers, and you're going to explain if you were surprised at the differences, and if you were, why, and if you weren't, why not. Then we're going to look at the differences between the channels, the gated channels and the leak channels. So we're going to go back to the checklist here. We've done 3 and 4. Now we're going to use the sliding tool to move across here and move back and forth and see what's happening as just before the peak, the sodium gated channel opens, and then just at peak, the potassium gated channel opens. Then just after, or right at the low point, the potassium channel closes, and then the gated channel closes. And then you're completing questions 5 and 4 with that. Okay, so just as we zoom in, we can see this happening. Opening up, see it opening, those gated channels, and then closing. And the potassium closes much sooner than the sodium. Okay, answering those 2 questions, what happens, what's the difference? And then you're going to do question number 7, which is using the graph that we just talked about. And then the last thing you're going to do is you're going to take 3 points on that graph, and you're recording the potential, membrane potential at rest, at peak, and immediately after. And that is how you do it.